Good morning, students. Today we are going to discuss about the culture media and its importance for the growth of different type of microbial culture. As we all know, culture is a term given to the microorganisms that are cultivated in the lab for the purpose of identifying and studying them. The purpose of identifying the bacteria is for the pathogenic involvement, antibiotic resistance, the culture and development of newly synthesized or identified bacterial cultures. To maintain and preserve the cultures of pure strains, we must implant the proper media required for their growth. Medium or the media is a combination of ingredients that will support the growth and cultivation of microorganisms by providing all the essential nutrients and factors required for the proper growth of microbial cultures. So, firstly we have to discuss about what is the culture media. Every living organism required certain type of food materials or the nutrients for their proper growth and reproduction. These food materials or substance required for growing microorganisms in vitro. It means outside the body of an organisms are called as culture media. The growth medium can be a solid, liquid or semi-solid as per the requirement of the medium or the microorganism's growth. Different types of media are used for different types of cells. Okay. Basically, the culture media that we imply are used for the following purposes like identifying pathogens, isolating and identifying the different type of organisms which are causing the diseases in human, in plants or animals, as well as they are used in the studying of antibiotic resistance, understanding microbial metabolism. As we all know that different type of microorganisms in plants or discuss as we discussed earlier that they have a different type of microbial metabolism. So to identify the microbial metabolism present in them, we must culture media as per their requirement. Contamination testing and the checking antimicrobial agents can be done with the culturing and maintenance of microbes. Now, we also estimate viable counts of the microorganisms. Viable counts means the number of living cells present in the microbial growth. The uses of culture media is to grow microorganisms, to identify the cause of infection, to study the characteristics of microorganisms as well as the biological products formation like vaccines, toxoids and antigens and some serums also. Based on the culture media importance and their functions, these culture medias are divided into following types like physical state. Based on the physical state, they are divided into the three types. Mainly is solid media, liquid media or semi-solid media. And the types based on the ingredients or the nutrients availability of the media, there are several types can be identified such as the simple media, complex media, synthetic media, selective media, differential media or enrichment media. As well as some microbes are aerobic in nature and some microbes are anaerobic in nature. So depend on their requirement, the media can also be divided into two types like aerobic media and anaerobic media. So we are going to discuss about this very shortly. That is, solid media is made up of nutrients composed or solidified using the agar-agar. Liquid media is a broth or the nutrient solution that is implant for the growth of microbes. Semi-solid media is a media which have a soft nature and which is mostly used to describe the bacterial motility and separating the motile bacteria from the non-motile strains. And based on the ingredients, as we all know, that is simple media, complex media, simple media examples are like nutrient broth, nutrient agar. Complex media are the media which contains the specific type of nutrients required for the particular type of growth of microbes, like blood agar, chocolate agar, peptones, meat extract and yeast extract are included in the complex media. Synthetic media is a media which is in which the nutrient content is specifically known to us. Fourth one is a selective media, subtype of a special media, and differential media. Selective media is the media in which certain type of food dyes or antibiotics are added to the media for the type of growth of particular organisms. Differential media are the media in which specific type of dyes are added or special type of metabolic substrates are added to incorporate the growth of microbes of particular color, which can be used as a identifying property. Examples are McConkey's agar, TCBS agar, XLD agar, etc. In enrichment media, the substrate to substrate, the unwanted organisms, mixed culture media are inoculated in it, which the particular type of enrichment substance is added to the media for selected growth of microbes. And the third classification based on the oxygen requirement is type of media such as anaerobic media and aerobic media. In this media, the chemicals or the nutrients required for the microbes to obtain their growth in anaerobic conditions are added. Examples are cooked meat medium or thioglycolate medium. And the last one is the aerobic media, in which the nutrients needed for the growth of aerobic microorganisms are added. These are